Welcome to What Did I Do in My Biochem Lab Yesterday, where I tell you guys what I did in my biochem lab yesterday. Yesterday was DNA ligation. So what that is, we had a bunch of plasmids from a bunch of E. coli bacteria, which is little circular bits of DNA, and then we cut them open, so basically we made them go from this to that. And then we got a bunch of DNA from the lambda virus, and then we got all that prepared. And then you just basically take the plasmids, add a bunch of stuff to it, take the DNA, add a bunch of stuff to it, leave them alone to cook for a little bit, wait for an entire hour for them to incubate and sit and do nothing for a while. Then when they're done cooking, take them out, add some more things to the plasmids in order to get the dephosphorylate them, which means take a phosphate group and cut them off in order to allow the ends to be a little bit better. Then we take the bacteria DNA plasmids and the viral DNA, and you add a bunch more stuff to them, put them in a centrifuge to get them all spun around nicely and get all the junk separated from the DNA, because we don't want like all the salts and the proteins and all that. We don't want that, we just want the DNA. So get the DNA separated out. Then once we're left with the nice clean DNA from the bacteria and the plasmids, we add them all together in a bunch of other chemicals that are supposed to make them stitch together. Those are called DNA ligases. So we get there's some buffer, we get the plasmids, the DNA, the ligase, bit of other stuff, put it all together, and you wait. What's supposed to be the end result is full circular bacteria plasmids again, which has the viral DNA stuck into the plasmids. So basically we cloned some viral DNA into bacteria plasmids. So now they're clones. Then, next week, we have the second part of this lab where we take the cloned plasmids and put them into real E. coli bacteria and see if they grew and see if we successfully cloned the DNA into the plasmids and if we have new, fun, mutant, transgenic bacteria.